We've been working on what is one of the most important diseases affecting livestock and humans across the bulk of Africa. The disease makes it impossible for many people to keep cattle and indeed goats and sheep. And the consequence of that is that their agricultural systems become very much harder to use. It's not just cattle, it's not just meat, it's not just milk, it's also traction for ploughing, it's also means of delivering goods to markets, it's about manure, and it's about an entire agricultural system which relies upon livestock. Trypanosomiasis is transmitted by the tsetse fly, and for many, many years people have been trying to control the disease by controlling the tsetse fly, by using drugs, by using sprays on animals, and trying to develop vaccines. All of these have problems. It's possible to control tsetse fly, but it requires sustained, intense, expensive activity. Drugs are relatively toxic and expensive for farmers. And people have been working on developing a vaccine for a long time and have made no progress whatsoever, because the trypanosome has this very clever trick of continually changing its surface coat and fooling the immune system. So as soon as the immune system mounts a response, the trypanosome changes its presentation. The cost of trypanosomiasis has been put into the billions of dollars to Africa. It's very difficult to quantify, but there's no doubt that the impact on agriculture and therefore human welfare is huge. And also livestock acting as reservoirs for human sleeping sickness is very significant. And we believe that if we can control disease in livestock, we can reduce transmission into humans. This project set out to look for a sustainable solution to trypanosomiasis, and it's based on the presence of animals which are highly resistant to trypanosomiasis, which occur in West Africa. The best example of resistant animals is the Ndama breed. Now, the Ndama is not a very useful cattle for agriculture. It's small, it's not very productive, but it's extremely resistant. So this project set out to find a sustainable solution to the problem of trypanosomiasis by understanding the basis of that resistance. Now, that's a very complex biological challenge, and in fact it's a classical challenge that's faced biologists in the post-genome era, the last 5 or 10 years. We rather naively thought that once we had genome sequences in humans and other species, then all of these problems would be instantly understandable. It turned out that once we had the genome sequence, that was only the start of a huge challenge to understand how that genome works, how the genes interact, when they're switched on, when they're switched off.